All right, I'm Todd Yates from UCLA, and it's my great delight today to have the chance to introduce the next speaker, a great colleague and friend in science, Jack Johnson from the Scripps Research Institute. So Jack grew up and went to college in the Midwest, and then he went to Iowa State to earn his PhD, where he studied inorganic chemistry and importantly studied crystallography, and there he became and remains a real expert in the methods of crystallographic calculations. Jack decided to leave finish his PhD and go do his postdoc and to continue with crystallography, but instead of trying to crystallize molecules that are composed of maybe 3 or 4 or 5 atoms, to crystallize molecules the size of viruses, and this was in the very early days. So Jack went to Michael Rossmann's laboratory and was the first one there as a postdoc to undertake virus crystallography there, working first on plant viruses and then on others. Jack's notable success there, I think, earned him a position, so he stayed on at Purdue and basically rose through the ranks there studying various virus structures and remained there for about 20 years. In the early 90s, Jack became the subject of a relentless recruiting effort by the Scripps Research Institute, where they would bring him out a month every summer to stay in La Jolla. And the reason I know that is because I was the beneficiary of the same condo the month before every summer I would go down. And the only rule about going there was I had to get me and my family out by the end of August, so Jack Johnson could take over the condo. And I remember asking the Scripps folks, isn't Jack going to get tired of coming out here, leaving Indiana and coming out to La Jolla every summer, such a long way? And they said to me, on the contrary, we think Jack's going to get tired of returning to Indiana if we bring him out enough times. And that scientific prediction turned out to be true. So I think in about 95 or 96, Jack came to Scripps permanently, and he's been there ever since. So at Scripps, Jack continued his prolific exploration and discoveries in all kinds of viruses, those that infect higher animals and plants and insects and tubules and archaea and all manner of organisms. And his work led to lots of interesting firsts and also to some important themes and surprises. Among the themes that came out of Jack's work was that viruses are actually quite dynamic and polymorphic, and the processes by which they go from nascent protein chains to mature viruses have many steps, and they're really interesting, and so that was an important theme. And also some surprises, and one of my favorite surprises that came from Jack's work was the discovery that the HK97 viral capsid is able to form an unusually thin shell because it adopts a topology where the chains are covalently linked in a way that creates chainmail in the surface of the capsid. So there were lots and lots of interesting things that came out of Jack's work, and then he also did and continues work repurposing viral capsids for different kinds of materials, so quite a diverse interest surrounding viruses. Besides Jack's own discoveries in viruses, he's actually been a champion of the virus structure field over the decades. He's written a number of review articles that have been influential to others, and his laboratory created and maintained the Viper database, which remains a resource, the most important resource in my view, for basically exploring virus structures and architectures and triangulation numbers and things like that. Jack has done his own important work in viruses and also drawn a number of others into the field, and I think probably our honoree today, Bill Gelbart, is probably among those many people who have been influenced by Jack's work, and so we're excited to have him here to tell us about his work on virus structure.
So is that hooked up with? Okay, well, thanks very much, Todd, for a nice introduction. I should say, whenever Todd talks about people involved with computational crystallography and so forth, he's one of the true heroes of this area, and as we've seen already, also protein design, and just a vast number of different things. And I have to reflect back on when I first had contact with Bill Gelbart. He sent me a large envelope of reprints that arrived without any prior knowledge. And my secretary put those on my desk, and I opened this up, and it had a lot of the theoretical types of things, and this was pre-Alex days. And anyway, I looked at this, and I thought, well, I don't know what I'm supposed to do with this. I don't even think there was a note with it, Bill. Anyway, he called me one day and said, "Did you get the package?" And I said, "Yeah, I think I know what the package you're talking about." And the bottom line is that that was probably 15 years ago, and right in the early 2000s. And being around Bill and his colleagues and Chuck Nobler have been one of the most stimulating things that I've been involved with. So I appreciate it very much, and I'm absolutely delighted to be here to talk in this symposium. So this is, Todd, you'll appreciate this. It's an oldie but goodie, but and it goes back to what Todd was referring to. Anyway, HK97 is the star of this presentation. It's a lambda-like phage. It has a capsid that's about 800 angstroms in diameter, and it has this very long tail. And for a virus crystallographer, working on bacteriophage was one of the holy grail studies, but as a crystallographer, these tails caused a great deal of problem in terms of crystallization. Of course, today with electron microscopy, we can ignore the tails and do lots of nice structural work in spite of the tails. But in order to do crystallography, we needed to have a solution of this problem with the tail. And the solution was to collaborate with Roger Hendricks and Bob Duda at the University of Pittsburgh, who were able to take just two genes, one for the capsid protein of the particle and a protease that's associated with the particle, both encoded by the virus, of course. And they took those two genes, expressed them in E. coli, and they could make particles that were, for all intents and purposes, identical to these particles, to the particles in the capsid, but without the tail. And that meant that we could grow crystals of these, and these crystals diffracted to 3.5 angstroms resolution, and we were successful in getting that structure. So being large particles, 900 angstroms in diameter, we had very large unit cells, very small order-to-order separation of the diffraction maxima, but with modern synchrotrons, we were able to resolve these individual diffraction patterns and work our way through and determine the structure. This is maybe one of the last structures of this size that will ever be done by crystallography, since cryo-EM has now been the method of choice. But it was a big job. There were 22 million reflections that we had to measure, and it gave us just under 5 million unique reflections, and we were able to determine the structure at 3.5 angstroms resolution. Okay, so we've seen some examples of particles, particularly from Don, and here we have classical organization of a virus particle, where we have the same subunit that's forming pentamers here and hexamers here. And this has a T equals 7, and the odd T numbers actually have a hand associated with them. So this is a T equals 7 LAVO structure, and what that tells us is that there are 7 capsid proteins in the icosahedral asymmetric unit, 6 in the hexamer and 1 in the pentamer, and then the rest of the particle is generated. So one of the first things that we did was to extract the subunits from each of these 7 positions in the capsid and superimpose them, and those structures superimposed with very, very high fidelity. In other words, there was very little difference in their structure depending on whether they were involved in hexamers or pentamers. Now, the fold of the subunit at the time that it was determined was novel. Nothing quite like this had been seen before. It's got a long, we called it the spine helix. It's got an alpha-beta structure that clusters around the hexamers and pentamers. And then it's got this long loop structure. And we'll come back to this in just a few minutes, but this the asparagine and this lysine are critical for the cross-linking that takes place during the maturation of this particle. Now, one of the interesting things about this fold was when we discovered it in about 2001, it was unique. And then over the years, there have been many more of these folds that have been discovered in other bacteriophages, and notably in herpes simplex virus, obviously a mammalian virus. So this same fold seems to be showing up a lot. And I was at a meeting last April where a guy named Matt Sullivan was there, and he showed this slide. And he says, furthermore, 4 of the 5 most abundant viral metagenome-derived protein clusters in the metaproteome represent capsid proteins containing the HK97-like protein fold previously found in many viruses that infect all 3 domains of life. The dominance of these proteins within our data set, as well as their global distribution throughout the world's oceans and seas, supports prior hypotheses that this HK97-like protein fold is the most abundant biological structure on Earth. So given the fact that we've got 10 to the 31 phages approximately, it's a very popular fold. Now, when we looked at the capsid structure that we determined, we were absolutely amazed because it looked very, very different from any other capsid structure that we had seen. All of these subunits were laying on top of each other. Each subunit was making contact with 5 to 9 other neighbors. And what we had seen in the past were well-behaved subunits where you had an envelope and those envelopes didn't overlap with each other. And this structure immediately told us why there had to be an elaborate maturation process, because you could not get to this organization of protein in one step. And if we look at the capsid itself, it was quite remarkable. It was only 18 angstroms thick, 660 angstroms in its maximum dimension, and yet it was extremely stable. So the stabilization occurred because of this cross-link between the lysine and asparagine, which is autocatalytic, with the release of ammonia. And these are cross-linking to other subunits, obviously. They're a long way apart in the same subunit. But what we were really impressed with was the fact that these created this chain-linked, chainmail fabric that holds these protein particles together, and the only way you can take them apart is to treat them with a protease. You can put them on a gel, and they just sit at the top of the gel because they are completely integrated. All of the hexamers and pentamers are integrated with each other, which is another reason why there must be a maturation process. You don't just reach that state of affairs in one step. So we then spent the next decade going through and looking at all of the maturation intermediates that we were able to generate. The structure I just showed you was the fully mature particle, what we called head 2. Now, the 2 genes that are expressed in Roger and Bob's system are the capsid protein and this viral protease. Now, the first assembly product is what's called ProHead 1, and that's a 19 megadalton particle, and it packages the protease. Now, normally, this is just a transient particle that you can't isolate, and because this protease cleaves off the first 103 amino acids from the N-termini of all of the subunits, and we end up with ProHead 1. And that's what we purify out of the expression system. Now, by making a protease, a non-functioning protease, we can actually purify this ProHead 1 with a protease bound inside but not active. We were able to do structures of a lot of these, structures of all of the intermediates that we could identify. And the ones that we're going to focus on is this ProHead 2 and the transition that goes out to the mature particle. Now, ProHead 2, the cryo-EM structure of ProHead 2, which was at about 20 angstroms resolution, immediately told us what the real initial assembly product looked like, because these subunits are forming hexamers, but they're not true hexamers. They're like two trapezoids that are skewed relative to each other, and then this is the pentamer. But when we looked at that cryo-EM structure, we could take the subunit structure that we got from our high-resolution work with the mature particle, and we could fit it in there. And now, when you look at those subunits, they're very well behaved. They have a nice envelope. Those envelopes don't overlap with each other, and it's a classic sort of assembly that we have seen in many other viruses. And if we look at the profile, that structure is very, very corrugated, and has a very different profile than what the mature particle has. So when the particle matures, it goes from this organization of protein to this organization of protein with a size change from about 500 angstroms in diameter to almost 700 angstroms in diameter. And the maturation that we look at in vitro is triggered by lowering the pH. That maturation occurs in vivo when DNA is pumped into these particles. So we could recapitulate in vitro this maturation process. Now, this is just a movie that shows how the subunits reorganize during this maturation process. All we did was morph our model in the ProHead 2 into the mature particle. And you notice we can see how these subunits are expanding relative to each other. And we also could see how the hexamers symmetrized during this process. So whoops, I'm sorry. That was a mistake. Let's see if we can get this thing going again. Anyway, this is, we'll just have to look at this one more time and hope I don't press the wrong button again. But anyway, we can see how much motion there is in these subunits. And then when we look at it from the side, we can see how the entire particle is expanding during this process. And notably, we can watch how the subunits change from being these asymmetric hexamers to being symmetric hexamers during this maturation process. And what's really interesting here when we look at this is what's happening to the residues that cross-link with each other, because they're not cross-linked in the ProHead. In fact, in the ProHead, if we zoom in, the lysine is sitting out in this position, the asparagine is sitting here, and they move more than 25 angstroms to have this juxtaposition for this autocatalytic reaction to occur. And if you look at something called a Hoberman sphere, we actually have a very, very neat mechanical model for what's going on here. Because in the procapsid, the subunits are more or less radial and well-defined. And then what happens is they go tangential, and the particle expands, and you get all of this overlapping of the subunits that occur during that process. So it's a very neat sort of organizational change that's completely spontaneous when you lower the pH from 7 to 5. Now, I want to just show a little more detail about the first transition that takes place between ProHead 2 and this expansion intermediate 1, as it's called, because we were able to characterize this by small angle X-ray scattering and by fluorescence studies. So the only thing you have to know about small angle scattering is that a big sphere generates a compact diffraction pattern, and a small sphere generates a broad scattering pattern. And the scattering pattern itself looks like these bullseyes. And so we just look at this kind of beading structure. And if you have a structural change during the transition, we have a very sensitive way to measure that structural change with small angle X-ray scattering. So what we did was we took our ProHead 2 particles and we measured the small angle X-ray scattering from those, which was in gold. And then we lowered the pH, and every 2 seconds we did an exposure, and then there was a 5-second interval, and we did the next exposure. And we see what were apparently all these intermediate structures between the ProHead and this expansion intermediate 1. Expansion intermediate 1 is larger, so it has a more compact pattern. But in fact, people like Todd know that when you see these knots in any kind of a data set where you're watching a transition, it means it's a two-state system. And all that we're watching is the progression of the number of particles in one state going to the other state. And there's really no intermediates that we're sensitive to. The analogy is popcorn. You put popcorn in a kettle, you heat it up, and you have two states. You've got the kernel and you've got popcorn. And that's what we're measuring here. We're watching the kettle fill up with popcorn. So Kelly Lee said, I want to look at the actual corn popping. And so he did this fluorescence experiment where he treated a slide with biotin, with PEG, with an occasional biotin on it. And then he took virus particles, and he also put a biotin-labeled PEG attaching to that. And then he mixed them and added some streptavidin, and he could immobilize these particles on the surface. And now we can watch the individual virus particles, which in this case were fluorescently labeled with Psi3, and we can see these particles as they exist on that surface. Now, what Kelly needed to do was to find a fluorescent probe that was sensitive to the two states of the particle so that he could watch the transition occur in vitro, one particle at a time, not as an ensemble like we did in the Sachs experiment. And he was able to do that with a probe called DBDS. And if you take the ensemble of particles labeled with that and you lower the pH, you watch the fluorescence increase in a linear way, indicating that this fluorescent molecule is actually reporting on the state of the particle. And now, when he did this with the individual particles, we could watch the actual transition. He put these particles on a slide, he lowered the pH, and now you could watch these particles transition as they lit up during the process. And we recapitulated essentially the Sachs experiment one particle at a time with this experiment. And if we look at the individual pixels, we can see the particles that have undergone the transition. But now we can go and look at the actual transition. We can see that it's a stochastic process. Some particles sit around for 60 seconds and then transition. Notice the transition itself just takes about 1 second. Other particles transition immediately. Some particles sit for a long time. So it's an exact recapitulation of what we saw in the Sachs experiment, where we watched the kettle filling up with popcorn, only now we're watching the individual particles fill up. And the most important thing that came out of this was that the transition itself was very fast and took depends on what scales we're talking about, but it's about a 1 second event. It's not slow. And this is the cryo-EM reconstruction of this so-called expansion intermediate 1 particle that is the larger particle. And already we can see that these particles have symmetric hexamers. So when that transition occurs, it goes from skewed to symmetric hexamers. And we also know from this experiment that these particles, that these ProHead 2 particles must be in an energetic state. And we believe then that energy is actually released when it goes through this process from prohead 2 to expansion intermediate 1. And then if we went back into the literature, and way back in 93, John King's lab characterized the amount of heat given off during this transition in P22, and it was about 21.5 kcals per mole. So these particles are energetically stabilized in some way, and then during the maturation, the energy is given off. So we were very, very interested in what the mechanism was for actually storing energy in this particle. And we felt that the only way that we could do that was to get a high-resolution structure of this prohead 2 particle so that we could see what was the difference in the subunit organization when these particles are in this skewed hexamer versus the symmetric state in the mature particle. So Ilya Gertzman in the lab had to do some molecular surgery on these particles in order to get them to crystallize. And we also needed to get a special mutant that Roger Hendricks and Bob Duda prepared that was what they called reluctant to mature. Because most of the time when we tried to crystallize things, we ended up with mature particles. So their reluctant to mature mutant allowed Ilya to be able to get crystals of this prohead 2 particle. And they diffracted to about 3.6 angstroms resolution. And when he got the structure, it was really quite informative. The prohead 2 structure had these subunits standing more or less vertical, which is how we modeled them into the cryo-EM. And then the head 2, they're laying more or less tangential. But most importantly, when he looked at the subunit structures, they really looked significantly different than the mature particles. This spine helix, which is very, very straight in the mature particles, is actually bent. And there's other features of this subunit that looked distinctly different than what we saw in the mature particle. And in fact, if we superimposed the what we called the p-loop region, we could see that there was a large conformational change of the individual subunits between the prohead 2 particle and this head 1 particle. And if we look at the morph of the 2 subunits from one to the other, we can see that there's a tremendous reorganization of the tertiary structure of these subunits that end up with this straight helix in the fully mature particle. So what our conclusion was that somehow or another these distorted tertiary structures were actually the energy storage mechanism for these prohead 2 particles. So then we have to ask, how does this happen? And when we look at the prohead 1 particles that have not been cleaved, these are what we call the delta domains. These are residues 1 to 103 that are on the inside of the particle and associating with each other, and they guide assembly. They're like a scaffolding protein that guides assembly of these particles. And am I at 5 minutes? 2 minutes? Oh, well, we're going to get back on schedule. If you actually delete these, genetically delete these, you don't get any assembly at all. So that N-terminal portion of these subunits is absolutely critical for proper assembly. And here's the model that we came up with. So we've got our subunits, and those subunits have these N-terminal regions, residues 2 to 103, and those guide the association of the subunits, and in the process actually distort the subunits as they move into this quaternary structure. We now complete the particle formation of this so-called prohead 1 particle but they're packaging protease. And we've shown that those protease molecules actually associate directly with the delta domain. So the protease molecules are bound to their substrate in this ProHead 1 particle. Then the protease cleaves off these delta domains, but the quaternary structure stabilizes the tertiary structure distortions that were imposed back here. And now we've got our energy-rich particle that's ready for maturation and does that in an exothermic way. So that's the sort of model that we have come up with for how you create an energy landscape with just 2 gene products. And I don't have time to tell you about all of the other kinds of experiments that we've done, but this is just sort of a summary. What I've been talking to you about at the latter part here is this motion from ProHead 2 to expansion intermediate 1. And this is the motion that takes place. Now, the next event that occurs is when we go from EI1, expansion intermediate 1, to something we call a balloon. And this is the motion that occurs during that transition. And then the final step that takes place is when we transition from the balloon to the finally fully mature particle, and that's a rather subtle reorganization of the subunits. But we've been able to map that entire process from initial assembly product to fully mature particle at near atomic resolution and with a variety of other biophysical methods. So what do we conclude? The HK97 scaffolding proteins, which are the delta domain, residues 1 to 103, interact with each other, distorting the subunit tertiary structures of residues 104 to 385, the rest of the subunit, and guiding the quaternary structure assembly. The quaternary structure then stabilizes this tertiary structure distortion, and the removal of the delta domains by proteolysis creates the metastable particle. Then packaging of a double-stranded DNA in vivo or lowering the pH in vitro drives the particle from this local energy minimum down the maturation cascade. So the entire process is driven by this energetically favorable profile. And then we think that this is probably a general process in the assembly of bacteriophage capsids, where usually you have scaffolding proteins that are guiding the assembly, but they get released just like the delta domain does, and that the quaternary structure of these first assembly products is stabilizing distortions in the tertiary structure. And in fact, we've seen P22 procapsids now, a different bacteriophage, and they have exactly the same distortions in the tertiary structure that we saw in HK97. So this was a project that involved a lot of different groups. Very sadly, in August, our sort of patriarch of this project, Roger Hendricks, died. But there have been a variety of people involved. Kelly Lee explained a lot of biophysics to me and did the fluorescence experiments. Lou Gann back here did later maturation events. Alistair Stephen here did a lot of the early cryo-EM experiments. And Ilya Gertzman back here was the hero that got the structure of the ProHead 2 that allowed us to put forward the model that we have today. So with that, I'll thank you for your attention and try to answer any questions.